Hello everyone and welcome back to ISCT TV. We're joined here with some of our amazing abstract winners who are going to talk to us a little bit about their work. Would you please introduce yourselves and tell us a little bit about your abstracts?
Sure, my name is David Wald. I'm a physician scientist from the Case Comprehensive Cancer Center and a small startup company called CureAI. My background is being trained as a pathologist, and then I have an academic research lab that's focused on cell therapy product development and translation to early phase clinical trials. The focus of my abstract was on trying to overcome some of the hurdles that are currently present with CAR T therapy. So even though CAR T therapy has worked remarkably well for at least a subset of malignancies, there's still a number of major challenges inherent in the field, such as poor accessibility of the product, high expenses of the product, high relapse rates, and toxicities that make accessibility and use of the product challenging, particularly in low and middle income countries outside the United States. And so the purpose of our abstract was to develop what we call an ultra-fast or a less than 1 day CAR T manufacturing process that's very simple, scalable, and it benefits by enriching in what's called the naive or early memory stem T cells, which impart biological functions to the product that make it have better proliferation and hopefully efficacy and durability in patients, as well as a better safety profile by having less cytokine release. And so we had recently completed a phase 1 trial and almost finished with a phase 1b trial where we've tested a version of a CAR T product with targeting CD19 using this ultra-fast manufacturing process, and we saw very promising clinical results, including the majority of patients undergoing complete remissions that have been durable and a very low toxicity profile. And so we're excited to try to further this work into a phase 2 clinical study.
Amazing, thank you.
I'm Christopher Duma. My day job is I'm a neurosurgeon at Hoag Hospital in Newport Beach.
Quite a day job.
Well, it actually led me to this startup. I was very tired of seeing patients come across my desk with Alzheimer's disease, ALS, MS, all these neurodegenerative diseases that we had absolutely nothing for. I figured there's got to be a different way, a different approach. And I've always been interested in stem cell work. I've dabbled in it earlier on in my career, which is a whole different story. But we decided to do some animal work. We did pig work, FDA submission, and in 2022, the FDA granted us the ability, which was my dream, to inject stem cells directly into the ventricles of the brain that are selected for their expression of Wnt. You're all very, I'm sure, familiar with the Wnt-catenin pathway. Wnt is a magical substance, really, that is very critical in the maintenance of our brain cells. The amyloid and the tau story. Wnt is critical in neural cell maintenance, and I figured if we could get the Wnt wave, to actually get going there in the brain, in that subventricular zone right next to the ventricle wall, maybe we could get some regeneration of neural tissue and better cell maintenance. So we're finishing our phase 1 trial. We did 6 patients, and that's what the poster was for. We were very happy to receive the award from ISCT for the West Coast submission, and really appreciate that we're appreciated by ISCT. So thank you very much for acknowledging our work.
Thank you all for submitting. It's because of people like you that we get to have conferences like this and that we get to advance the field. We're lucky to have you all with us.
Yeah, so thanks. My name is Caroline Rulleau. I'm finishing actually my PhD at the University of Montreal. And I'm working in the laboratory of Dr. Jean-Sébastien Delille, so in the Maisonneuve-Rosemont Hospital. For this work that we submitted, it's about the successful redirection of the TCR specificity using healthy blood donors to redirect specificity against cryptic leukemia antigen. So we did that using a non-viral TCR engineering with a one-step CRISPR-Cas9 based approach. We did an orthotopic replacement of the TCR, and using this method, we were successful to find 2 TCR that could be actionable for leukemia immunotherapy. So now we're looking to continue to find more TCR that we could use for immunotherapy and to extend that to more HLA type and more malignancies. So that's it. And for the award, I received the Early Stage Professional Award, so I was very grateful to have received this award as well.
Something that I find amazing is that you all do such different work, and yet you all were able to come to a conference like this. So I was wondering, why ISCT? Why submit your work here?
So, so actually this is my first time to ISCT, but I've really enjoyed the experience.
Welcome.
I'm definitely going to come back again. And I think for me the reason to come here is, working in the cell therapy space, it's a very multidisciplinary space that takes people from all different backgrounds to come together, and you need to work as a team. And so this type of conference, one, it teaches us what's out there outside of our local center, and also brings us connections with people to work with outside our center. So I've been very fortunate to meet a lot of potential new collaborators through.
Well, I'm glad you're enjoying your first experience with us.
Yeah, I agree with that. Our thrust to come here was the stem cell part of this whole thing, right? And we have been around the world already in Madrid and Pittsburgh, Philadelphia, et cetera, at clinical meetings. Alzheimer's Association International Conference, the Alzheimer's, clinical trials, CTAD, clinical trials for Alzheimer's disease. We've been to all of those, and frankly, have not gotten the same, embrace that we've got here at this stem cell meeting. So I think it's going to get out there a little bit more, but just being in this element, it really, I think we were chosen because it's very disruptive work. I mean, we're outside the box, and we're going someplace that no one has ever done before. This is first in human work.
Right.
Yeah, so similar to my colleague answer, so it's my second time at ISCT. I went last year, was my first time, and I thought it was very nice because you have people from different backgrounds. So for projects like that are super multidisciplinary, we know we're gonna have we're gonna be able to talk with people with a very fundamental science background. But for us, for example, looking to translate to GMP grade settings, we can also exchange with different companies. So that's a very great conference for that.
Well, thank you all so much for being here with us, both here at ISCT TV and just here at ISCT in general. I really enjoyed getting to hear all of you speak earlier. It was absolutely incredible. So thank you again for joining us. It's been an honor. So
Thank you.
Thank you.
thank you all for tuning in. We'll see you next time.
Thanks very much.